To process information at the subatomic level, human engineers build elaborate dilution refrigerators. These complex machines drop temperatures down to fractions of a degree above absolute zero. They encase the processing chips inside extreme vacuums, sealing them off from any stray magnetic fields or ambient radiation. This extreme isolation prevents decoherence. If a delicate quantum particle encounters even the slightest thermal heat or molecular noise, its wave of probability instantly collapses into a fixed, rigid state. Now look inside a living human cell. It is a microscopic hurricane. Organelles and proteins violently collide in a hot, wet, highly pressurized environment. Classical physics dictates that fragile quantum states cannot survive here. The heat and friction should destroy them in a fraction of a millisecond. Yet researchers are finding them anyway. Biology is actively hacking the quantum realm, forcing us to reconcile the messy reality of life with the rigid laws of subatomic physics. Subscribe to join us at this new intersection of biology and mechanics. In classical chemistry, a reaction requires kinetic energy. It behaves like a physical object trying to roll over a steep hill. If the object lacks the momentum to reach the peak, it rolls back down, and the reaction fails. Human metabolism routinely violates this model. Enzymes like dihydrofolate reductase, or DHFR, process chemical reactions millions of times faster than classical kinetic energy calculations allow. They achieve this by treating the hydrogen proton not as a solid particle, but as a fluctuating wave of probability. This is quantum tunneling. Instead of climbing the energy barrier, the wave stretches and instantaneously teleports through the physical obstacle, appearing on the other side. To facilitate this, the enzyme physically compresses the barrier, shortening the distance the proton travels. Your metabolism operates by bypassing the laws of classical kinetics entirely. We see a different kind of quantum engineering inside a plant leaf. When a single photon of sunlight strikes a leaf, it creates a highly energized packet called an exciton. Now, this exciton has a singular goal. It must navigate a dense, chaotic maze of pigment molecules, known as the antenna complex, to deliver its energy to the plant's reaction center. If this energy moved like a classical pinball, it would bounce randomly off the walls of the maze. By the time it finally found the exit, it would have lost massive amounts of energy as waste heat. In 2007, researchers analyzing photosynthetic bacteria at UC Berkeley discovered a rhythmic anomaly inside these cells. They called it quantum beating. The plant forces the exciton into a state of quantum superposition. Instead of a single particle bouncing through one corridor, the energy spreads out as a wave, exploring every possible path through the maze simultaneously. The exact millisecond the wave touches the most efficient route, the superposition collapses. The energy instantly commits to that single optimal path. By sustaining quantum coherence at room temperature, plants achieve near-perfect energy transfer. Human engineers struggle to maintain this kind of efficiency inside million-dollar freezers. These subatomic mechanics also operate inside the central instruction manual of human life, our DNA. The Watson-Crick base pairs, the rungs of the DNA double helix, are held together by delicate hydrogen bonds. Recent studies from the Leverhulme Center show that the hydrogen protons within these bonds don't always stay firmly in place. They frequently quantum tunnel across the gap, teleporting to the opposite strand. Normally, the proton drops back to its original position. But if the DNA strands split for replication while the proton is tunneled to the wrong side, the code breaks. This incorrectly bonded state is called a tautomer. When the cell tries to read it, it creates a spontaneous genetic mutation, permanently altering the code. Every time this copying error propagates, the organism changes. The slow, overarching march of human evolution is driven by microscopic fluctuations in quantum probability. The strangest implications of quantum biology point toward the origin of human consciousness itself. Physicists and neuroscientists developed the orchestrated objective reduction, or ORC-OR theory, to explain how the physical brain processes subjective experience. Deep inside the brain's neurons are structural tubes called microtubules. Within these tubes, clouds of delocalized pi electrons float freely through the tubulin protein. In theory, these electrons enter an entangled stage. They act as biological qubits, calculating massive amounts of sensory information simultaneously across the neural network. When this vast superposition reaches a critical mass, it instantly collapses. That precise moment of collapse is theoretically experienced as a single flash of conscious awareness. By decoding the specific quantum triggers behind DNA mutations and the subatomic speeds of enzyme catalysis, we gain the tools to design drug therapies that target disease with molecular precision. These discoveries are providing the first templates for new, sustainable materials that mirror the high-efficiency energy transfer of a plant leaf. The chaotic, boiling interior of a living cell poses no obstacle to quantum physics. Over 2 billion years of evolution, nature has actively engineered that chaos to protect, harness, and run entirely on the subatomic realm. Deep inside every cell in your body, enzymes are tearing apart and rebuilding molecules millions of times per second. This rapid-fire chemical processing keeps our metabolism running, but it happens at a pace that has puzzled researchers for decades. Calculations show that these reactions happen so fast, classical chemistry alone struggles to explain them. The enzymes appear to rely on a quantum shortcut called tunneling, where particles pass directly through energy barriers. If the subatomic world is driving something as basic as digestion, the same influence likely extends to the very center of the cell. This leads us to the nucleus, where the genetic blueprint is stored in the double helix of DNA. While we often view this structure as a stable physical ladder, the atoms making up these base pairs are in a state of constant microscopic flux. The genetic code functions as a fluctuating subatomic system, shifting constantly beneath the surface of the double helix. The two strands of the DNA helix are held together by hydrogen atoms, specifically single protons acting as the chemical glue between base pairs. Because they are so small, these protons are subject to the laws of probability. At room temperature, they can behave like spread-out waves, existing in multiple overlapping locations at once. Occasionally, a proton will vanish from its normal position and instantly reappear on the opposite DNA strand. It doesn't move across the gap, it tunnels straight through the energy barrier. If the DNA helix unzips for replication while that proton is misaligned, the cell's machinery creates a mismatched base pair. This results in a tautomer, a structural error that hard-codes a mutation into the new strand of DNA. This subatomic flux introduces a layer of uncertainty into the replication process, providing a quantum mechanism for the spontaneous mutations that have shaped biological history for billions of years. This subatomic influence extends to how we interact with the environment. Traditional chemistry explains our sense of smell through molecular geometry, where an odorant's specific physical shape must dock into a matching receptor to trigger a signal. However, molecules with entirely different shapes can smell identical to us. Even stranger, molecules with the exact same shape, but containing heavier isotopes, trigger completely different scents. The quantum vibrational theory proposes receptors measure the specific frequency of a molecule's chemical bonds. The receptor waits for an electron to tunnel across a gap. It completes the jump only if the odor molecule provides the correct vibrational energy, completing a circuit. Olfaction functions as a biological sensor for subatomic vibrations, translating the energy signatures of molecules into the scents we perceive. Neuroscience remains divided over the mechanism that allows biological cells to generate subjective internal experience. Physicist Roger Penrose proposed that the brain exploits quantum mechanics to perform operations beyond the reach of classical neurobiology. Deep inside our neurons are hollow cylinders called microtubules, built from protein subunits called tubulin. Penrose suggested these proteins can exist in two different structural states simultaneously, acting as quantum bits or qubits. Because these proteins contain delocalized electrons, they can become quantumly entangled. This creates a unified network of superposition across the brain, holding millions of possible computations in a delicate, unresolved state. The Orchestrated Objective Reduction Model, or ORCH-OR, suggests that this entangled state builds up until it reaches a specific physical threshold, at which point the entire system rapidly collapses. This theory posits that the moment of collapse is what we experience as a single moment of human awareness. Critics point out that delicate quantum states usually require extreme cold and perfect isolation to survive. In the warm, wet environment of the body, they break down through a process called decoherence. Biological systems have had billions of years to engineer solutions to this problem. Evidence suggests that evolution has actually developed molecular structures capable of shielding these quantum reactions from the surrounding thermal noise. By mimicking these biological shields, researchers can now simulate subatomic molecular interactions with high accuracy. This allows for the design of targeted drug therapies that interact with the specific quantum properties of a patient's cells. These same principles are being used to build quantum sensors. These devices can detect disease biomarkers at concentrations far below the limits of traditional laboratory equipment. Understanding the quantum mechanics of life is enabling a wave of precise medical interventions that classical chemistry alone could not map. When we map the interconnected structures of neural networks alongside the sprawling filaments of the cosmic web, the visual similarities suggest a deeper physical link. The processes that power our cells and spark our thoughts rely on the same subatomic mechanics that dictate the behavior of photons in stars. Human biology operates within a continuum of physical laws, where the same quantum rules governing the universe dictate the behavior of our own internal systems. Thank you for watching. As research continues to unfold, quantum biology is providing a new lens through which to view the mechanics of life. We'd love to hear your thoughts on Penrose's theory of consciousness, so drop a comment below. And if you enjoyed this journey into the subatomic world, please like this video and subscribe to the channel for more deep dives into science's mysteries.